This lecture is going to cover the physiology of the sympathetic nervous system. Other lectures had already covered the anatomy of the sympathetic nervous system, and there'll be other lectures that cover the anatomy and physiology of the parasympathetic nervous system. So we're going to look at the physiology of the sympathetic nervous system. So the preganglionic neurons of the sympathetic nervous system is these guys are relatively short, and they release at their endings, nerve endings, acetylcholine. Now notice again, here's the cell body, or the soma, of the preganglionic neurons, which are located within the central nervous system. And again, specifically within the lateral great horns of the thoracic and parts of the lumbar spinal cord. So T1 to L2. The preganglionic neurons are going to be found along within the white rami communicans. Then they synapse with the cell bodies of the postganglionic neurons, either in associated, the ganglion could be part of the sympathetic trunk ganglion, the collateral ganglion, or the adrenal medulla. But these all release acetylcholine. On the postganglionic neuron, the receptor that binds to acetylcholine are always nicotinic receptors. Nicotinic receptors are ligand-gated ion channels, specifically allowing a lot of sodium to move across. Now these are always going to be excitatory. So if you have enough acetylcholine released, you'll get an action potential. And then we'll stimulate an action potential that will travel along the postganglionic neuron to its target, which could be smooth muscle, cardiac muscle, gland, adipose tissue. Now in the green, the neurotransmitter released by sympathetic endings, most of them, it is norepinephrine. We're always going to have a few exceptions. When we talk about the few exceptions, we'll make a point of it. But most of them will release norepinephrine. There'll be some that we'll talk about specifically today in that acetylcholine is released. Now these fibers that release norepinephrine, we're going to refer to them as adrenergic fibers. If you have adrenergic fibers, they're going to release norepinephrine. We also have some instances in which the neurotransmitter release would be nitric oxide. Over here on the left, this is trying to represent what we'd have in the adrenal medulla. So the preganglionic neuron synapses with this modified sympathetic ganglion found in the adrenal medulla, and what we have is in the green we release norepinephrine, and in the red it releases epinephrine. So these two hormones are then released and travel into the bloodstream, where they can have similar effects as if we had direct innervation by the nervous system, as long as you have specific receptors at the target structure. So and the receptors are going to vary, so that's one of the things that we have to talk about. So again, cell body of the preganglionic neuron located within the central nervous system. Cell bodies of postganglionic neurons are located within a ganglion. The postganglionic neurons are going to be, for the most part, in the sympathetic division, are going to be fairly long, while the preganglionic neurons are fairly short. All preganglionic neurons release acetylcholine. They bind nicotinic receptors. The majority of the postganglionic neurons release norepinephrine, with a couple exceptions. Now the receptors for epinephrine and norepinephrine are examples of what we call metabotropic receptors. They are not ion channels, like the nicotinic receptor, which is an ion channel. The metabotropic receptors, the specific type that we'll see with the receptors for epinephrine and norepinephrine, are G protein coupled receptors. And what happens is when the epinephrine or norepinephrine bind to its receptor, it's going to cause changes within the cell in its metabolism, where then it will either open up particular channels in the membrane or inhibit, depending on what particular type of receptor that's involved. So it's an indirect effect. So the nicotinic receptor would be a direct effect, because when acetylcholine binds that receptor, it opens up an ion channel, because it is an ion channel. Now there are multiple subtypes of receptors for epinephrine and norepinephrine, and we refer to them as adrenergic receptors. And we have alpha 1, alpha 2, beta 1, beta 2, beta 3. Now the reason why we have different ones, this gives you specificity. There's different, or each receptor may, some may be excitatory, some may be inhibitory. They're going to be localized in specific tissues, so it gives some sort of specificity to it. So you can have epinephrine or norepinephrine causing one effect in a particular organ, causing a different effect, or not even have an effect on an organ, all depending upon the receptors involved. Now, the alpha 1 adrenergic receptors are going to be excitatory. These are going to be found in most tissues. So most tissues will have alpha 1 adrenergic receptors. Now, how they elicit their effect is through, again, it's these signal transduction pathways. It has an indirect effect, and it actually does it by increasing the levels of calcium through its effects on things called IP3 and DHE, which you don't really have to write down. But the alpha 1 adrenergic receptors are going to be found in most tissues. They're going to be excitatory. The alpha 2 adrenergic receptors, their effects on the targets will be inhibitory. They exert their actions by decreasing cyclic AMP levels. You're going to find these in the central nervous system, the peripheral nervous system, platelets. For the most part, we are not going to be discussing these today, so you can pretty much ignore those. The beta 1 adrenergic receptors are excitatory. They work via increasing cyclic AMP levels. And where we're going to find the beta 1 adrenergic receptors are going to be in the heart, the liver, the kidney. What you're going to notice today, the only example I'll give you is the heart. So I'm going to show you how to time will limit how much memorization, because I'm only going to give you examples of its effects on the heart. The beta 2 adrenergic receptors are inhibitory. These receptors are going to be located within smooth muscle of vessels of the heart and skeletal muscle. So blood vessels that serve the heart and skeletal muscle. It's also going to be found within smooth muscle of the lungs, the intestines, pretty much just GI tract. You can put GI tract, and bronchi. We'll just put GI tract in general. So smooth muscles associated with it. Beta 3 adrenergic receptors are found in adipose tissue. And what this does is it increases the breakdown of fat, lipolysis. So it's stimulatory. That's the only place that we're going to see the beta 3. So we're going to look at various target structures and look at the effects of sympathetic activity on these target structures, but we'll also be adding what type of receptors are involved. Now, it's important that you know what type of receptors are involved, because this is an important target for pharmacologics. A lot of pharmacologics are used to target specific adrenergic receptors. So it's going to be important that you understand which receptor is located in which target organ. And if you know what the effect is, normal effect, if I block it or I use a drug that binds to it and has a similar effect as epinephrine or norepinephrine binding to it, you'll know what the effects are. The one thing about the sympathetic nervous system is they have referred to as you have what we call mass activation because of this divergence and convergence. You don't see this as much in the parasympathetic nervous system. Because the preganglionic neurons branch in synapse and ganglia at any level. So these can come up here, come down here, number of different levels that you can have one presynaptic neuron that synapses on multiple postsynaptic neurons at multiple levels, so up to about 20. Then also at different levels, you can have several preganglionic neurons synapse on one postganglionic neuron. So that would be convergence. Divergence is that preganglionic neuron branching off and synapsing with multiple postganglionic neurons. Convergence is you got one preganglionic neuron that synapse, or multiple preganglionic neurons that synapse with one postganglionic neuron. So you can have this mass activation. You don't really see that much with the parasympathetic nervous system. Also the effects of the sympathetic activation are much more longer lasting than you see with the parasympathetic. So the effects last a little bit longer. Another thing is once you did what you need to do, you pretty much have to terminate the effects. You have to break down the various neurotransmitters or epinephrine released by the adrenal medulla. Is to terminate the effects, there's a number of different ways to do it. Is the neurons can take the neurotransmitter back up. You don't even have to break it down. Sometimes we'll just take it back up and we'll just keep it for another time. You also have a couple enzymes that can break down various neurotransmitters. So catechol-O-methyltransferase and monoamine oxidase both can break down norepinephrine or epinephrine, but they can also break down such things, or in regards to catechol-O-methyltransferase, that can break down dopamine. Monoamine oxidase also breaks down serotonin. And the reason why I want to mention that is that there are medications called COMT inhibitors, which are used to treat Parkinson's disease. So Parkinson's actually, with treatment of Parkinson's disease, they give them multiple things at one time. Is Parkinson's disease, remember, they have a problem with dopamine levels, and they have insufficient dopamine levels. So they give them something called levodopa, which is like dopamine, and that is a substrate for this enzyme. Well, we don't want to break it down. We want to prolong the effects in that subject. And so what they'll do is they'll also give them a COMT inhibitor. There's also evidence that inherited variants of this enzyme here are linked to people with schizophrenia. So there is, and with people with schizophrenia, there's issues with certain neurotransmitters. Now, monoamine oxidase inhibitors are actually used to treat depression. And the thing is with people who take things like monoamine oxidase inhibitors, they have to watch what they eat, because if they eat things or drink things that have a lot of something called tyramine in it, what can happen is they have, as a result of inhibiting this enzyme that breaks down things like norepinephrine, epinephrine, is they can have a significant increase in norepinephrine levels, which can cause, say, example, a hypertensive crisis where their blood pressure gets really high. So that if you're interested in it, you could always look up these various substances because they're kind of interesting. Now, what we're going to do first is we're going to look at structures that are innervated by the sympathetic trunk ganglion. So we're going to look at various structures innervated by looking at the receptors involved in the various effects. So we're going to be looking at, for sympathetic trunk, remember we're looking at structures associated with the skin, blood vessels, and the eye, and the thoracic cavity. So what we're going to do first is let's look at blood vessels. Now, it's going to be different depending where those blood vessels are located. If the blood vessels are in the skin, the gut, or the kidneys, these guys have alpha-1, and I'm not going to write it out, but they have, this is the only time I'm going to write it out, adrenergic receptors. Is alpha-1 adrenergic receptors, remember if you go back to the previous couple slides, is these are going to, so let's actually go back so you can see where I was writing it in there. So alpha-1, remember, are excitatory. So let's see, what does it mean by excitatory in regards to smooth muscle? So with this, what it's going to do is going to cause smooth muscle to contract. And a result, the smooth muscle found in blood vessels is going to result in vasoconstriction, which would reduce blood flow. But blood vessels found in skeletal muscle and cardiac muscle, they have beta 2 adrenergic receptors. And so these ones were inhibitory. So what it does is it causes the smooth muscle to relax, which really results in vasodilation. These are protected circulations. So I want you to think about, okay, think logically when you're trying to figure out the effects of the sympathetic nervous system. You're in fight or flight mode. What you need is you need their blood to go to more important organs. You want it to make sure it goes to skeletal muscle if you're going to run or fight. You want to make sure you have plenty of blood supplied to the heart. You're going to divert it away from places that you really don't need it. I don't need it in the gut. I can worry about digesting my food later. Skin, no. Now kidneys, now I do want blood to the kidneys, but it will divert some blood away from the kidneys when you're in that fight or flight mode. But the kidney, when you look at kidneys, this is going to be real important in cases of hemorrhage. You're going to try to prevent, you want to make sure blood goes to the heart and the lungs and the brain. You're going to divert away from the kidneys. But generally, sympathetic stimulation, blood vessels, most of the blood vessels that we talk about have the alpha-1, so you're going to get vasoconstriction. But we refer to some of the sort of the protected populations, like in cardiac muscle, the smooth muscle in those organs are going to relax and you get vasodilation. Now there is a little aside though, is there's not a lot of innervation to skeletal muscle to begin with. Most blood flow in skeletal muscle as well as cardiac muscle is controlled not by the nervous system, by local metabolites. But of what we have there, we do have more beta 2 adrenergic receptors. Another target, where this would be associated with your skin, is the erector pili muscle, which is smooth muscle. When the erector pili muscle is stimulated, it is actually via the alpha-1 adrenergic receptor, which you said was excitatory or stimulatory. What you get is piloerection. In piloerection, this is what causes the hair to stand up on end, which leads to goosebumps. Now the purpose, when we talked about it in previous lectures, is piloerection. What it does, the hair will trap a layer of air next to the skin, which is then warmed by the body and by body heat, and it's used as an insulating layer. It's not a huge factor in humans because we don't have that much hair. In animals, it also gives them, when they have the piloerection, they look intimidating, which again, when you're in fight or flight mode, it's like you're ready to fight, you're trying to intimidate another animal. That hair standing on end is going to be intimidating. But the piloerection is, we talked about it's important in temperature regulation. And the sympathetic nervous system is going to be important in temperature regulation. The next target would be adipose tissue. So you do have a lot of adipose tissue in the hypodermis, but you have in other places in the body. Is adipose tissue, you have the beta 3 adrenergic receptor. What that does is it triggers the breakdown of fat, and what you get is, what you'll have is fatty acids that will go out into the bloodstream and can be used as energy. So I want you to think about when you're in fight or flight mode, you're going to need some energy to make ATP in order to run, to fight, and so you need to break down, in this case you're going to break down fat. So you have lipolysis as a result of the sympathetic nervous system. This also will play a role in temperature regulation too, especially if you talk about brown fat and what you see in babies, but you don't have to worry about that. Other structures associated with the skin are our sweat glands. So here's going to be a little different one. The sweat glands, the fibers that innervate it, are not adrenergic. They're cholinergic. So the postganglionic neurons release acetylcholine onto the sweat glands, and the receptors are muscarinic receptors. So this one's going to be kind of important. This is kind of like the odd man out. So the cholinergic receptors release acetylcholine, and they bind to muscarinic, and it's actually we call an M1 muscarinic receptor. And as a result, you increase the amount of sweat that you're producing, which is important in temperature regulation. But when you're in fight or flight mode, you tend to sweat. When you're stressed, you sweat. That helps to cool off the skin. So this one is kind of unique in that we don't have an adrenergic receptor involved because it's acetylcholine that will be the neurotransmitter. So those are structures associated with the skin. Now what about structures in the head, like the eyes? Now the iris of the eye has two muscles associated with it. And the muscle that we're going to be looking at is the muscle that will be active when you're in sympathetic mode, is this outward muscle right here. And this is your pupillary dilator muscle, otherwise known as the pupillary radial muscle. This smooth muscle, when it's stimulated to contract, is going to increase the diameter of the pupil, allowing more light into the eye. So once you think about it, fight or flight mode, you want to make sure you let as much light in your eyes so you can see kind of stuff as you're moving along from a distance, say if you're running from something. This is also actually important during sex too, and you get an increased pupil diameter during sex. Is this one, because it's stimulatory, it's muscles contracting, it's mediated through the alpha-1 adrenergic receptor. Now the cranial nerve, and it's a cranial nerve that's carrying these fibers, is going to be cranial nerve number 3, the oculomotor nerve. So the preganglionic neurons emerged from the thoracolumbar spinal cord. It traveled up the sympathetic trunk, where it then synapsed within the superior cervical ganglia, and then the fibers are actually brought along cranial nerve number 3 to this particular muscle in the eye. Next structure, these are the only ones we're going to do with the head, is we're going to look at structures in the thoracic cavity. We're going to look at the lungs and then the heart. So the lungs, you have some smooth muscle associated with the bronchi and bronchioles. And the diameter of the airways can determine the amount of airflow. Is when you're in fight or flight mode, think about, you now to make sure you get enough air coming in and out of your lungs when you're, say, running or exercising. So what you want to happen is you want these airways to get bigger, so it allows more airflow. And the way you do it is you have bronchodilation, which is mediated through the beta-2 adrenergic receptors. So the beta-2 adrenergic receptors, they are inhibitory. So what they do is that release of norepinephrine is going to cause the smooth muscle to relax, which causes the airways to open up. So you can get dilation of the bronchi, but also dilation of the bronchioles, allow more airflow. Now in the heart, the sympathetic innervation to the heart, this has more than one effect. As you'll notice is we have innervation to these structures here. Now this structure right there is where your pacemaker of your heart. It will control heart rate. So one of the things it does is it increases heart rate. Now don't worry about this one. It's something we'll talk about in AMP2, so I don't want you to worry about that yet. And it also innervates pretty much most of the muscle, pretty much all the muscle in the heart. And this one's an important one in the ventricles. They're the ones that are going to be, when they contract, pumping blood up to the heart, up to the lungs or the rest of your body. So you have an increased contractility, which increases something we call stroke volume. Stroke volume is the volume of blood that you're able to get out of the heart with each heartbeat. So an increase in heart rate, increase in stroke volume, this is mediated through the beta-1 adrenergic receptors, which you find within the muscle and on those structures such as the SA node or the AV node. So you can have effects in rate because of its effects on those nodes, or you have effects on contractility because of how it affects the muscle. So these guys are excitatory. So the beta-1 adrenergic receptor is excitatory. So overall what happens is your cardiac output, and this is the abbreviation for cardiac output, your cardiac output goes up when you're in sympathetic mode, which is important when you're running or doing any type of exercise or any type of exertion. You want to make sure you get enough oxygen and nutrients to different tissues. So this is the end of structures that are where the ganglia are part of the sympathetic trunk or the paravertebral ganglion. So we're going to look at target structures innervated by the celiac ganglion. So we're actually going to do one. I could do other ones, but I'm only going to pick one, and that's the liver. In the liver, the liver has an alpha-1 adrenergic receptor, and in the liver what it does is it triggers the breakdown of glycogen. So glycogenolysis. Glycogen is just stored glucose, and when you break it down, now you have glucose available for tissues to use for energy. So again, think about it. When you're fight or flight mode, you want more energy. So you need this to make ATP. So this is going to be important when you're in that fight or flight mode. Now the other things that are going to be innervated is actually I'm going to mention just a couple real quick, because it's, even though it's not in your notes, is the celiac ganglion is going to be the location of cell bodies of postganglionic neurons that innervate the stomach also, and the liver, the gallbladder. So the overall thing, when you think digestive system, I want you to think overall the sympathetic nervous system is inhibitory. You don't really care about digesting food or any of that when you're in fight or flight mode. So what it does to the stomach is it decreases motility. It decreases secretions in the stomach. Other things that will affect are various sphincters, which control exit and entrances to different components. So you'll notice I have various sphincters labeled, and as a rule, what it does to sphincters within the GI tract is it causes those sphincters to contract. So you can kind of somehow put that down there. Now the sphincter of Oddi is a sphincter. It's also called the hepatopancreatic sphincter. This controls the exit of pancreatic enzymes as well as bile to go into the small intestine. So it keeps it closed. So again, you're not worrying about digestion when you're in fight or flight mode. Now the other things we'll see is the small and large intestine. Now you see here, here's small part of the small intestine, large intestine. That is again part of the digestive system. So let's see what it does in there. So the small and large intestines, all of the small intestines will be innervated by the superior mesenteric ganglion. Large intestines only the first couple parts. So up here and here, this is called the ascending colon. This is the transverse colon. This is descending. Sigmoid, here's the rectum and anus. Superior mesenteric pretty much does these two. Inferior mesenteric ganglion will go here on down. So the latter parts of the large intestine. Now overall, again, what I had mentioned, sympathetic nervous system on the digestive system is inhibitory. It will decrease motility. And the same thing right now would apply to the stomach that I had mentioned earlier. So decrease motility, decrease secretions from those regions, and that's via beta-2 adrenergic receptors. So remember, beta-2 adrenergic receptors are inhibitory. You find them in smooth muscle in a lot of locations in the body. But it causes contraction of sphincters. That's alpha 1. So remember, contraction of sphincters, you're causing that muscle to contract. So in order for that to happen, those have alpha 1 adrenergic receptors. So sphincters contract, things aren't going to be moving, leaving different parts of the GI tract. Things aren't moving, or you're not having a lot of secretions, also because of the sympathetic nervous system. So here, you've got, just want to notice you have a sphincter here, ileocecal valve, which controls the movements of things from the latter part of the small intestine, which is called the ileum with a U, into the colon. And here's some other ones I do want to mention. There's an internal and external anal sphincter. The sympathetic will only have effects on the internal because this is smooth muscle. The external sphincter is skeletal muscle, so that's innervated by somatic motor fibers. So the internal sphincter, anal sphincter, would be stimulated to contract by the sympathetic nervous system. Other structures innervated by the sympathetic nervous system. Now these guys are all coming from the inferior mesenteric ganglion. So we have the urinary bladder. Now we're going to look just at the red fibers. The green is going to represent somatic nervous system. The blue represents the parasympathetic nervous system. So the urinary bladder does have some innervation by the sympathetic nervous system. What it does is it's going to inhibit contractions, because you're not in rest and digest. When you rest and digest, yeah, that's when you're going to want to go to the bathroom. So inhibit contractions via beta 2, and it also causes contraction of the internal, oops, spelled it wrong, internal urethral sphincter, which is smooth muscle. So this is a picture showing you in a male. So the internal sphincter, anal sphincter, is right here. Here's the external, and this one is innervated by somatic fibers coming from the pudendal nerve. Now the internal sphincter is innervated by fibers coming from the pelvic sphincteric nerves, which be carrying parasympathetic fibers, but we're worried right now about sympathetic fibers. So here the sympathetic innervation is going to cause that to contract, which again is alpha 1 adrenergic receptor. So what you'll notice is a common theme. Alpha 1 adrenergic receptors, wherever they're located, those are going to be stimulatory. They're going to cause those muscles to contract. The beta 2, they were inhibitory. They're going to cause muscles to relax. The beta 1 we saw, I only used that as an example, was in cardiac muscle. That was stimulatory. Now the other structure we're going to look at that has innervation from the inferior mesenteric ganglion is the genitals. So we're looking at a male. But you'll notice all the different structures, accessory structures, and of the male. So we have vas deferens, which is carrying sperm that was made here in the testes, stored right here, but it's going to smooth muscle associated with this structure. We have smooth muscle associated with various glands. Those are innervated by sympathetic fibers. And what happens during an orgasm is you have a process called emission. Emission is that movement of the sperm and various glandular secretions, which starts happening up here, along here. It's still within the male's body. Ejaculation is when it leaves the body. So the process of emission and ejaculation is due to sympathetic nervous system stimulation, and it's via alpha 1 adrenergic receptors. Similar thing in the female reproductive tract. So we do have sympathetic fibers found within the female reproductive tract, and that will be involved in orgasms, as in males. Now the last thing is the adrenal medulla. So the adrenal medulla, remember in that here, this is a modified sympathetic ganglion, is we have, when that is stimulated, it releases epinephrine and norepinephrine into the bloodstream. That has multiple effects. So all it is with the adrenal medulla is you release norepinephrine and epinephrine into the bloodstream, they will have the similar effects as what we saw in everything else that we talked about, as long as there's a receptor for it. Okay, so if there's a receptor, then it will bind to it and have an effect. So it'll have similar effects as the previous. So this is going to be the last part of the physiology, of the sympathetic nervous system.